Chapter 12. We all know that each individual organism exists because of the process of reproduction. So, let us study more about this process in the chapter Life Cycle. Observe the above pictures and answer the following questions. Which life process is shown in the pictures? Is it an essential life process like nutrition, respiration or excretion? Reproduction. All living organisms produce new individuals of the same species, that is, a new generation of the species from an existing individual. This fundamental characteristic of living things is known as reproduction. Reproduction is necessary to maintain the number of individuals of a species and to prevent their extinction. Let us look at one activity. Observe the picture and identify the plants belonging to the same species. Plants A and D belong to the same species. Plants B, C and E belongs to the same species. Organisms belonging to the same species look similar to each other and create new individuals that look like themselves. A basic process in reproduction is the creation of a DNA copy. Cells use chemical reactions to create copies of their DNA. In this process of copying, each time some variations take place. Thus, DNA copies generated are similar but not identical to the original. What is the importance of variations? Changes in ecological system which are beyond our control, like varying temperature, varying water level, can wipe out a species. But if variations in some individuals are suitable for new environment, there is a chance of survival of that species. Observe living things in figure. Observe different plants and animals around you. Do they reproduce in the same manner? No. All plants and animals do not reproduce in the same way. What are the modes of reproduction? Two main modes of reproduction are asexual reproduction and sexual reproduction. Asexual reproduction. Only one parent is involved in asexual reproduction, in which cells divide mitotically. Since there is no fusion of two different cells, the daughter cells produced by asexual reproduction are genetically identical to the parent cell. Lack of genetic variations is the disadvantage, while rapid reproduction is the advantage of asexual reproduction. Mitosis. It is a type of cell division leading to growth and development. It occurs in somatic cells. Diagrammatically, it can be represented as parent cell 2n divides into 2 times 2n daughter cell. Let us look at the modes of asexual reproduction used by unicellular organisms. Activity. Observe a permanent slide of amoeba showing binary fission under a compound microscope. Allow some aquatic vegetation to decompose in a jar of pond water. In this stagnant bacteria-rich environment, paramecium will often multiply, forming a ring just below the water surface. Let us see what is meant by binary fission. It is a method of asexual reproduction that is employed by most prokaryotes, some protozoa, and some organelles within eukaryotic cells. In it, the living cell divides into 2 equal or nearly equal parts which have potential to grow to the size of the original one. The plane of cytoplasmic division is through any plane in amoeba, which is simple binary fission. However, in some organisms, fission occurs through a specific axis, transverse or longitudinal binary fission. Now we will study multiple fission. During unfavorable conditions, the amoeba withdraws its pseudopodia, becomes almost round and secretes a hard covering called cyst. Inside the cyst, nucleus divides into many nuclei by repeated division, followed by division of cytoplasm. As a result, many daughter cells are formed. The cyst bursts to release the daughter cells during favorable condition. Budding. Yeast reproduce by budding, in which a small outgrowth is formed on the parental cell. The nucleus of the parental cell divides and one daughter nucleus migrates into the bud. The bud increases in size, separates, and grows further. Activity. Arrange the following plants and animals in ascending order with respect to their structural complexity. Plants: bacteria, sunflower, fungus, funaria, spirogyra. Answer: bacteria, fungus, spirogyra, funaria, and sunflower. Animals: frog, amoeba, planaria, human being, hydra. Answer: amoeba, hydra, planaria, frog, and human being. In unicellular organisms, all the life processes occur in a single cell, while multicellular organisms have different cell types which perform different functions. They have special organs placed at a definite place in the body, which are formed by specific tissues. In such organized organisms, method of cell-by-cell division to reproduce is impractical. Therefore, they show complex ways of reproduction. The complexity in reproduction increases with the complexity in structure of organisms. Let us look at the asexual reproduction in multicellular organisms. Activity: collect slimy green filaments from a pond. Put 1 or 2 filaments on a slide. Observe the slide under the microscope. It looks like this. Fragmentation. When water and nutrients are available, spirogyra grow and multiply rapidly. The filament of spirogyra undergoes fragmentation, resulting in numerous filaments. With cell enlargement and subsequent mitosis, each fragment grows and develops into a mature filament. Regeneration. The capacity to regenerate is very high among some animals. They can reconstruct the entire body from the isolated body cells. Regeneration is carried out by specialized cells. These cells proliferate and make large numbers of cells, which later develop into various cell types and tissues and helps in production of new organism. For example, when planaria is cut into many pieces, each piece develops into a whole planaria. This process occurs only if the planarial body gets cut into pieces, but animals cannot wait for being cut up to reproduce, so regeneration is not the same as reproduction. Budding. Organisms such as hydra use regenerative cells for reproduction in the process of budding. When hydra reaches maturity and is well fed, its body wall begins to form a rounded growth from the stalk of the adult. This growth is called a bud, which develops in time into a miniature hydra. The body layers, body cavity, and the digestive cavity of the young hydra are continuous with that of the parent hydra. The young hydra gets nourishment from the parent. When the young hydra is sufficiently developed to take up an independent existence, the base of the new hydra seals off and thus allows the new individual to break off from the parent hydra. Activity. Observe the figure and identify parts of plants involved in reproduction in each picture. Roots, leaves, and bud are the organs involved in reproduction. Vegetative propagation. When new plants are produced from the vegetative parts like roots, stems, leaves, and buds, it is known as vegetative propagation. For example, New plants of potato develop from eyes, buds on potato. Bryophyllum reproduces from the bud on the leaf margin. The root of sweet potato gives rise to new plants, etc. All plants produced by vegetative propagation are similar to the parent, as they are produced from a single parent. Plants produced by vegetative propagation take less time to grow and bear flowers and fruits earlier than those produced from seeds. Let us do this activity. Take a piece of moist chapati or bread. Leave it in a moist, warm place for 2 to 3 days. Observe the fungus grown on it. Spore formation. The hyphae of bread mold Rhizopus are thread-like structures. The mold forms spores inside a sporangium. When the spores are ready to leave the sporangium, it breaks open. If they land in a moist place, they germinate to form new mold. Cloning. Animal cloning is the process by which an entire organism is reproduced in a genetically identical manner from a single cell taken from the parent organism. This means the cloned animal is an exact duplicate in every way of its parent. It has the DNA which is identical to parent DNA. For example, Dolly sheep was the first mammal to be cloned from an adult cell rather than an embryo. Let us move to the next topic, sexual reproduction. Two parents are involved in sexual reproduction, one male and the other female. The main two processes involved in sexual reproduction are 1. Meiosis. It is a process in which halving the number of chromosomes from 2n to n takes place, resulting in the formation of haploid gametes. 2. Fertilization. It is a process in which the male gamete fuses with the female gamete, resulting in the formation of diploid zygote. It restores the number of chromosomes, that is 2n. Since there is fusion of two germ cells, offspring produced by sexual reproduction are different from parents. Variations give rise to variety and diversity. Variation enables organisms to adapt and survive in the changing environment. It helps to prevent the complete extinction of animal and plant species. Meiosis. It is a type of cell division leading to the production of gametes. It occurs in reproductive cells. Activity. Students, try to complete the table to get the difference between asexual and sexual reproduction. Sexual reproduction in plants. Flower is a functional unit concerned with sexual reproduction. You know that sepals, petals, stamens, and carpels are different parts of a flower. Carpel, female reproductive part of a flower, present in the center of flower, made of 3 parts, stigma, style, and ovary. Stigma, sticky terminal part of the style, receptive organ on which pollen germinates. Style, elongated part of carpel bearing stigma at its tip. Ovary, the swollen lower part of carpel containing 1 or more ovules. Each ovule has an egg cell, female germ cell. Stamen, male reproductive part of a flower, made of 2 parts, anther and filament. Anther, usually bilobed and produces pollen grains. Male germ cells are produced by pollen grains. Filament, stalk of anther. Pollination, the process of transfer of pollen grains from anther to stigma is called as pollination. If this transfer of pollen occurs in the same flower or another flower of the same plant, it is known as self-pollination. On the other hand, if pollen is transferred from one flower to the flower of another plant of the same species, it is known as cross-pollination. The agents of cross-pollination are wind, water, or animals. Activity. Try this in your garden. You have to shake the anthers of a flower onto the stigma of another flower to induce pollination between flowers of the same plant. Horticulturists use a special kind of brush to transfer pollen between the flowers of different plants. In the absence of honeybees, the yield of sunflower goes down tremendously. Why? Honeybees sit on the sunflower for nectar. When they sit on one flower, pollen gets stuck on their body. When these honeybees sit on another flower, these pollen grains get transferred. Fertilization. After the pollen grains land on stigma, it germinates. The pollen tube grows out from a pollen grain. It travels through the style to reach the ovary. Each pollen tube contains 2 male gametes and release them near the egg. One male gamete fuse with the egg cell to form zygote. The second male gamete fuses with a secondary nucleus in the embryo sac to form endosperm. This is called as double fertilization. The zygote develops into embryo and the endosperm serves as nutritive tissue for the growing embryo. This embryo is capable of growing into a new plant. Activity. Soak few seeds of green gram in water. After 7 to 8 hours, drain the excess water. Keep the seeds in a wet cloth for a day. Open a seed carefully and observe the different parts. Germination. After fertilization, the zygote divides several times to form an embryo within the ovule. The ovule develops into a seed and ovary develops into the fruit. The seed contains the future plant. It develops into the seedling under appropriate condition. This process is known as germination. How reproduction in human beings takes place. Observe the table showing human developmental stages. The male reproductive system. Testis and penis are the main reproductive organs of male reproductive system. Testis produces sperm's male germ cells. As formation of sperm require temperature lower than the normal body temperature, testes are located outside the abdominal cavity in the scrotum. Testes secrete hormone testosterone, which brings about changes in boys during puberty. Epididymis. Immature sperm travel to the epididymis for development and storage. Vas deferens. It is a passage through which the sperm travel towards the urethra. Seminal vesicle and prostate glands produce ejaculatory fluid which help the sperm in transport and provides nutrition. Penis. Portion of reproductive system that deliver the sperms to the site of fertilization. Sperms. A sperm cell is composed of a head which carries the genetic information, a middle part which carries mitochondria required for energy production, and a tail which is like a flagellum to help in movement towards the female germ cells. The female reproductive system. Vagina, uterus, oviduct, and ovaries are the main reproductive organs of female reproductive system. Vagina. It is a muscular tube that extends from the vaginal opening to the uterus. It provides the route for the menstrual blood to leave the body during menstruation. It is a pathway through which sperms enter into woman's body and is a pathway through which a baby comes out of the woman's body during childbirth. Uterus. Uterus is a muscular organ. Due to strong muscles and ability to expand and contract, the uterus can accommodate a growing fetus and can push the baby during labor. Oviducts connects uterus to the ovary. Ovaries. Two oval-shaped organs lie to the upper right and left of the uterus. They develop and release eggs into the oviduct. Secretes hormone estrogen, which brings about changes in girls during puberty. When a baby girl is born, the ovaries already contain thousands of immature eggs which remain inactive till maturity. On reaching puberty, a woman's ovaries usually release one egg each month. Menstruation. When the egg is not fertilized, this unfertilized egg along with blood and mucus form a flow which leaves the uterus. This is called menstruation, which lasts from 3 to 5 days. At birth, the female ovary contains approximately 2 to 4 million eggs. After birth, new eggs are not added. Out of these eggs, only about 400 are destined to be ovulated, while all others degenerate during development so that only a few remain by the age of 50. Thus, eggs ovulated near the age of 50 are 30 to 40 years older than those ovulated just after puberty. This aging of eggs can cause defects in children born to older women. Fertilization, development, and birth. The sperms enter through the vaginal passage, travel upwards, and reach the oviduct where they may encounter the egg. As soon as it is fertilized, the egg cell, zygote, begins to divide until it becomes a ball of cells called a blastocyst. This ball then implants itself in the wall of uterus. The development of embryo takes place inside the uterus, where the embryo gets all the nutrients and oxygen from its mother's blood in the oviduct supplied through the umbilical cord. From 9 months onwards, the baby is ready to take birth. It moves down. During birth, the cervix gradually opens and the baby is released through vagina. How twins are born. Sometimes a single fertilized egg may get split and separated into 2 parts during the early stage of cell division. Each part then develops into a separate individual. As these 2 individuals are produced from a single zygote, they look similar and are called identical twins, either both boys or both girls. Sometimes 2 eggs are released from ovaries simultaneously and get fertilized to produce 2 individuals. As these 2 individuals are produced from different zygotes, they are dissimilar, either both boys or both girls, or one boy and one girl. Population facts. The population of world has reached 7 billion in 2011. With the present trend of population growth, India is likely to overtake China and be the world's topmost in population by the year 2025. Students, do you know that July 11th is observed as World Population Day? Needs for and methods of family planning. There is undoubtedly a strong relation between high national fertility rate and measures of poverty. As the population density increases, decrease in per capita income and natural resources takes place. General health also goes down. It creates an economic burden on the nation. Large families affect both the individual as well as community life. Economic pressure, mother's poor health, children neglected at home, poor housing, malnutrition, insufficient medical care, lack of better education are some of the disadvantages of large family size. Limited family is advantageous to the individual as well as to the nation and ultimately to the entire human race. For better future of mankind, the rate of growth of human population must be reduced. Another problem related to human reproduction is the spread of sexually transmitted diseases, STDs. STDs spread from an infected person to a healthy person by unprotected sexual contact. Syphilis, gonorrhea, AIDS are some of the common sexually transmitted diseases which affect sexual health of human beings. To protect individuals from these STDs, sex education and preventive measures are necessary. Use of condom not only protects individuals from STDs but also helps in population control. Contraception to avoid pregnancy and to keep sexual health can be achieved by the use of condoms, oral pills, copper T, and some other methods. The WHO has prepared guidelines and considered reproductive health as a fundamental human right. Well, that ends the chapter. Hope it was a joyful learning. You have the options to view a mind map for a quick recap and a question and answer section to study the exercises. Wish you all the best. Bye till we meet again. Chapter 13. Physical traits of an individual are maintained because of the genes. Let us try to understand various facts about these genes in the chapter Mapping Our Genes. Observe the plants grown in a rose garden or a sugarcane field. Are the plants grown alike or do they look very different? All the plants grown in a rose garden or a sugarcane field look alike. Take the example of your class. Do all the students in your class look alike? No, all the students in the class do not look alike. Let us see what is meant by inheritance. We have already studied that reproduction is a process which gives rise to new individuals that are similar to their parents with subtle differences. Organisms that reproduce by asexual mode exhibit minor variations, whereas when the mode of reproduction is sexual, the variations are more. In this lesson, we shall deal with the mechanism by means of which variations are created and are passed on from one generation to another, or in other words, inherited. These inherited variations get accumulated in the individuals of forthcoming generations. Members of a generation inherit the characters from the members of the previous generation. This inheritance provides the general basic design of the body with some minor changes in it for the next generation. What would happen when this new generation would reproduce? The second generation would have differences that they inherit from the previous generation, and they shall also have certain newly created differences. The original organism at the top will give rise to individuals similar in body design but with minor differences. Each of them in turn give rise to individuals with minor differences. Some of those differences will be unique, others will be inherited from the respective parents. The above figure shows how diversity is created over the succeeding generations. The organism at the top gave rise to 2 organisms, and each of them gave rise to 2 each. When we observe these 7 individuals, we see that they are all similar in body design but are with minor differences. The figure represents the situation when the mode of reproduction is asexual. In asexual mode of reproduction, the individuals produced are very similar to one another and are with minor differences because of small inaccuracies involved in DNA copying. But when the mode of reproduction is sexual, then greater diversities will be generated. This can be explained when we discuss the laws of inheritance. We have all heard of the word heredity. Let us see what is meant by heredity. Heredity is defined as the transmission of traits, physical or mental, from parents to offspring. The offspring of dogs are dogs, pigeons are pigeons, humans are humans only. Inherited traits. It is a well-known fact that offspring do resemble their parents, but yet there are many differences between them. This resemblance and difference is the result of heredity. Let us know more about Gregor Johann Mendel. Johann Mendel, July 20th to January 6th, 1884, was an Augustinian priest and scientist who gained posthumous fame as the figurehead of the new science of genetics for his study of the inheritance of certain traits in pea plants. Mendel showed that the inheritance of these traits follows particular laws, which were later named after him. The significance of Mendel's work was not recognized until the turn of the 20th century. The independent rediscovery of these laws formed the foundation of the modern science of genetics. Let us study Mendel's law for inheritance of traits. The laws for inheritance of traits in human beings are based on the fact that both the mother and father contribute equal quantity of genetic material to the child. As the contribution by each parent is equal, then what will be the traits seen in the child? Mendel worked on these lines and formulated the main laws for such inheritance. It is very interesting to study the experiments conducted by him over a century ago. Mendel's experiments were based on a number of visible contrasting characters of garden peas, Pisum sativum, like tall or short plants, red or white flowers, round or wrinkled seeds, axial or terminal position of flowers, green or yellow-colored pod, etc. Mendel chose pea plants with different characteristics, like plants bearing red flowers and plants bearing white flowers for his experiments. He produced progeny, offspring, from them and calculated the percentages of plants with red or white flowers. Mendel's monohybrid cross. Mendel, during the tenure of his experiments, made several crosses of pea plants involving only one pair of contrasting characters or traits. Such crosses were termed as monohybrid crosses. We shall consider Mendel's cross of red-flowered and white-flowered pea plants as an example to study and understand the monohybrid cross. The parental generation P1, red-flowered and white-flowered pea plants, were used as parents for crossing, and thus they are said to be plants of P1 generation. Mendel used terms like dominant and recessive for red flowers and white flowers respectively. Dominant character is the one which masks or hides its recessive partner. Red flowers were called dominant because in the following generation all the plants were with red flowers. White flowers were termed as recessive as this character was not exhibited in the next generation. Mendel stated that the factors responsible for these traits were present in pairs. Today we call these factors as genes. Dominant genes are represented by capital letters and recessive genes are represented by small letters. As the genes occur in pairs, those for red flowers are written as RR and those for white flowers as RR. The pair factors or genes separate or segregate out during the formation of gametes in the P1 generation. Thus, two types of gametes are produced, one with R factors and the other with R factors. The following generation, that is, the generation produced by P1 generation, is called as the first filial generation or F1 generation. The first filial generation F1. Mendel observed that all the flowers of F1 generation were red in color. He realized that the red-flowered plants of F1 generation were different from the red-flowered plant of P1 generation, as the F1 generation plants had white-flowered parents. From the result of F1 generation, Mendel concluded that red was dominant over white. Though F1 individuals were bearing red-colored flowers, they contained factors for white-colored flowers too. Thus, the plants of F1 generation are phenotypically red but genotypically hybrids. The phenotype is the appearance or any detectable characteristic feature of an individual like red and white flowers. The genotype is a term that refers to the genetic composition of an individual. Thus, we can say that F1 red-colored plants are similar to P1 red-flowered plant in their phenotype but different in genotype. Red-flowered plants in P1 generation have RR genotype and thus produce only one type of gamete, R. The F1 red-flowered plant possess RR genotype and thus produce two types of gametes, namely R and R. Mendel continued with his experiments and allowed the F1 plants to self-pollinate in order to produce second filial generation F2. The second filial generation F2. The F2 generation has both red-flowered and white-flowered plants. Mendel found out that out of a total of 929 pea plants, 705 were red flowers and 224 plants were with white flowers. Thus, the phenotypic ratio is approximately 3 red is to 1 white. The genotypic ratio would be 1 RR is to 2 RR is to 1 RR, which is based on genetic constitution. Thus, it can be inferred that phenotypically there are two types of F2 plants, that is red and white flowered plants, but genotypically there are three types, namely A. F2 pure dominance, RR produced red flowers, homozygous. B. F2 pure recessive, RR produced white flowers, homozygous. C. F2 hybrid reds RR produced red flowers, heterozygous. With the help of a table, show Mendel's monohybrid cross of tall TT and short TT pea plant. Mendel's monohybrid cross of tall TT and short TT pea plant is shown in this way. How will you find out that F1 generation has traits both for tallness and dwarfness? Answer: plants of F1 generation were tall. Tallness of F1 generation was different from the tallness of plants of P1 generation, as the F1 generation plants had dwarf parents. From the result of F1 generation, it can be concluded that tallness was dominant over dwarfness, but had traits of both tallness and dwarfness. Write the phenotypic as well as the genotypic ratios. Answer: phenotypic ratio is equal to approximately 3 tall is to 1 dwarf. Genotypic ratio is equal to 1 TT is to 2 TT is to 1 TT. Now let us study Mendel's dihybrid cross. A dihybrid cross involved two pairs of contrasting characters. Mendel continued with his experiments on pea plants. This time he crossed pea plants having round and yellow seeds with plants bearing wrinkled and green seeds. We can see that here two pairs of contrasting characters, namely seed shape and seed color, is involved. Since this experiment involves two pairs of contrasting characters or traits, it is known as Mendel's dihybrid cross, parental generation P1. Mendel obtained pea plant with round and yellow seeds and plants bearing wrinkled and green seeds. When the gametes of the P1 generation are formed, each pair of genes, RR or YY, segregates independently of the other. Thus, the RRYY plants would produce only RY gametes and not RR or YY gametes. Similarly, the RRYY plants would produce RY gametes. Thus, we conclude that in gametes, a pair of genes is represented by only one of its members, first filial generation F1. On the basis of the experiments with monohybrid crosses, Mendel anticipated that the F1 generation plants would be with round yellow seeds. His prediction was right. Though the genotype of the pea plants were RRYY, but phenotypically They were plants with round yellow seeds, as yellow color is dominant over green and round shape over wrinkled. The F1 plants are called dihybrid. The F1 plants produce 4 types of gametes. They are RY, 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 out of which RY and RY are similar to the P1 gametes, hence known as parental combinations, and the remaining 2, that is RY and RY, are termed as recombinations. When F1 generation plants undergo self-pollination, they give rise to second filial generation F2. The 4 types of male gametes and the 4 types of female gametes give rise to 16 mating combinations, which are shown in the Punnett square or checkboard diagram. The male gametes are arranged on the top of the board and the female gametes on the side. It should be noted that the proportion of different types of F2 individuals are represented on the Punnett square and not their actual numbers. Mendel produced 556 pea plants, of which 315 had round yellow seeds, 101 were wrinkled yellow seeds, 108 were round green seeds, and 32 were wrinkled green seeds. Second filial generation F2. Observational study of F2 generation gives the results as shown. Phenotypic ratio is equal to 9:3:3:1. Let us do this activity. The above 4 phenotype involved 9 different genotype. Observe the figure and complete the following table. Allot numbers to the squares. The completed table is as shown on the screen. Write the genotypic ratio. Answer. Genotypic ratio is equal to 1 RRYY is to 4 RRYY is to 1 RRYY is to 1 RRYY is to 2 RRYY is to 2 RRYY is to 2 RRYY is to 2 RRYY is to 1 RRYY. Which squares show pure dominant and pure recessive traits? Answer. Squares of genotype RRYY shows pure dominant and genotype with RRYY shows pure recessive traits. Answer the questions. What do you mean by DNA? Answer. DNA means deoxyribonucleic acid. What is the peculiarity of its structure? Answer. DNA is a long structure made from repeating units and 2 helical chains, each coiled round the same axis. Name the scientists who put forward the most popular model of DNA. Answer. James D. Watson and Francis Crick put forward the most popular model of DNA. How do particular traits or characteristics get expressed? Let us find out the mechanism of heredity. How does it function? We are all aware of the fact that cellular DNA is the source of information for synthesizing proteins in the cell. A fragment of DNA that provides complete information about one protein is referred to as gene for that protein. How are these proteins related to the traits in plants? For example, let us consider tallness as a trait. We know that in plants, growth hormones are present. The height gained by plants depends upon the amount of growth hormone produced by the plant. The quantity of hormones produced by the plants depends upon the efficiency of the concerned enzyme. Efficient enzymes produce hormones in greater quantity, resulting in the plants gaining height and thus becoming tall. On the other hand, if the enzymes are less efficient, they produce less quantity of growth hormone, which results in the stunted growth of the plants. The interpretations of Mendelian experiments that we discussed earlier in the chapter suggest that during sexual reproduction, each of the parent contributes equally to the DNA of the progeny. Both the parents must be contributing a copy of the same gene. Thus, each pea plant have two sets of all genes. Thus, each gamete has only one gene set. How do gametes or germ cells make a single set of genes from two sets? If the progeny plants inherited only one gene set from each parent, then the experiment of dihybrid cross will not be true, as two traits, namely R and Y, would then be joined to each other and cannot be independently inherited. But it does not happen that way, because the fact is that each gene set is not present as a single long thread of DNA, but as separate independent pieces, each called as a chromosome, one each from the male and female parents. Each gamete takes one chromosome from each pair, and when two such gametes combine, they restore the normal number of chromosomes in the progeny. This is the mechanism of inheritance, and it also explains the results of Mendel's experiments. Do you know how sex is determined, male or female, a boy or a girl? In sexual reproduction, individuals of the either sex are involved. Let us look at the factors for sex determination. In most of the organisms, there is a genetic or chromosomal mechanism for determination of sex. Some organisms depend entirely on environmental factors for this. Thus, in some animals, the temperature at which the fertilized eggs are kept decides the sex of the offspring. How is sex determined in human beings? Sex determination in human beings is genetical. In other words, we can say that the sex of an individual is determined by the genes inherited by it from its parents. Earlier, we studied that similar set of genes are inherited from the parents. In that case, how can genetic inheritance determine sex? Out of the specific number of chromosome pairs that each species has, one pair decides the sex of the individual. This pair is referred to as sex chromosome. The number of chromosomes for a particular species is specific. In human beings, there are 46 chromosomes or 23 pairs, out of which 22 pairs are autosomes and one pair is sex chromosome. In human males, two dissimilar chromosomes are present, longer X and shorter Y. In human females, similar longer X chromosomes are present. This figure shows that chance for the child being a male or female is 50%. All children inherit X chromosome from their mother. Thus, the sex of the offspring is determined by the chromosome that they inherit from their father. If it is X, then the offspring will be a daughter, and if Y, then it will be a son. Students, it is interesting to know that the opposite condition, like that of humans, also occur, in which males have XY chromosomes and female have XX chromosomes. Here, males are heterozygous and females are homozygous. Example, birds and lepidopterans like moths and butterflies. Here the gametes from the female decides the sex. The ratio is not affected. Various special cases and exceptions also occur. Sex reversal is possible in certain fishes, amphibians, and birds. Let us now study how evolution took place. When we look around us, we see a variety of organisms. As the environment changes, the organisms also change many a times. These organisms, whether plants or animals, are well adapted to their environment. These organisms have certain salient characteristic features in them which help them to get well acclimatized to their environment. Those organisms which cannot adjust to their surroundings have no right to survive, is the rule of nature. Earlier in this chapter, we have discussed at length about the mechanism by means of which variations are created. These variations are then passed on from one generation to the next generation. The long-term effects of accumulation of variations make their study very interesting. Do these variations found in species have equal chances of survival in the environment in which they belong to? The answer is no, because depending upon the difference in the nature of variations, different individuals would have different advantages. For example, a bacterial species which can withstand excessive heat will survive well in conditions like a heat wave. Selection of variants by the environmental factors forms the basis for evolutionary processes. There are more than 1 crore species of animals found on this earth today. They are of varying size and complexity. Say from minute unicellular amoeba or paramecium to huge and gigantic whales. Likewise, from unicellular chlorella to the big banyan tree, there are about 2.5 crore species of plants on this earth. Life is present on all parts of the earth from equator to the poles in the air, soil, sea, wood, rock, etc. From time immemorial, man is curious to know about the origin of life on this planet and how the great diversity in it came into existence. Till date, many theories have been proposed to explain the origin and diversity of life on this planet. Out of all these theories, the theory of the evolution of life is the most widely accepted. Theory of evolution of life. According to the above-mentioned theory, a life substance appeared on the earth in the seawater. During course of time, this living substance developed into a unicellular form of life. Then some gradual and orderly changes took place in this unicellular form, and it developed into multicellular forms of greater and greater complexity. Then behavioral patterns also gradually underwent many changes. These changes were very slow and sequential. It took a prolonged period of nearly 300 crores of years. These changes and development of living things took place in all aspects, and that gave way for many different kinds of living things to come into existence. Hence, this process is called the evolution of life on Earth, which is also referred to as organic evolution. Organic evolution is thus the progressive development of animals and plants from ancestors of different forms and functions. Evidence for organic evolution. Organic theory of evolution is the most accepted theory as it answers many questions like how life came into existence, how various forms developed, and how more competent species come into existence. Various types of evidence are also available to support the theory. In spite of many adaptive differences in protoplasm and cells and their manifestations of life, processes like growth, reproduction, metabolism are essentially the same in all organisms. Let us answer these questions. Observe the pictures and answer the following question. What are the common features that you observe among plants and among the animals? Answer. The common features among plants and animals is that all the three plants have green leaves and all the animals have a body covering of fur. Your answer indicates that there are similarities among the three, and thus it suggests common origin and evolution from a common ancestry. Let us see what is meant by anatomical evidence. Generally speaking, the human arm, a bull's leg, a bat's wing, and whale's fin have nothing in common. See this figure. They all perform different functions in their respective animals. Since their functions are different, there is difference in their structures. When we observe the figures minutely, we are able to establish similarities in the bones of these different organs and their joints. This similarity is attributed to having developed from a common ancestor. Students, it is interesting to know that organisms have organs of similar function. If they are fundamentally unlike except in function, they are termed as analogous. Example, tail fin of lobster and flukes of whale, wings of housefly and wings of a bird, eyes of arthropods and vertebrates. Organs which are fundamentally the same in structure but perhaps modified for widely different functions are termed as homologous. Example, the forelimbs of man adapted for handling, bat and bird adapted for flying, whale and seal adapted for swimming have the same principal skeletal composition. The scales of an ovulate pine cone correspond to the carpels of a flower, and the scales of a staminate cone correspond to the stamens of a flower. Similarities observed between the homologous organs are due to the fact that evolution leads to modify the existing structures for new modified functions, creating a new structure. What is meant by vestigial structures? Some organisms possess non-functional structures which in other organisms have essential functions. Vestigial organs are the non-functional organs. Man does not possess a tail like a monkey. However, he does have a rudimentary tailbone composed of 4 fused vertebrae in the same position. There are also some rudimentary muscles attached to the tailbone. These muscles are like that of the muscles of the monkey, which it uses to swing the tail. This evidence advocates the fact that monkey and man have common ancestry. Thus, anatomical study of the bodily structure of plants and animals too supports the theory of evolution. It is interesting to know about vestigial organs. Ill-developed structures or structures which are of no use are sometimes found in both animals and plants. They are referred as vestigial organs. The same organ may be functional in one organism but vestigial in another. Vermiform appendix of man is one such organ among others. The reason could be human ancestors were eating uncooked food with considerable cellulose content in them. The normal function of cecum and appendix in mammals is the digestion of cellulose. Man shows a number of other vestigial structures. About 100 have been named. A few examples are ear muscles, wisdom teeth, plica semilunaris, representing nictitating membrane of the eye, coccyx, reduced tail, etc. In plants, the scale-like leaves on the Indian pine, a plant which has lost its chlorophyll and become saprophytic in nature, are vestigial. There are certain flowers in which stamens bear no anthers. What are fossils? How are they formed? Let us see. Paleontological evidence. The process of fossilization takes a very long period of time. In order to study the fossil, the land is excavated. Fossils are collected from different levels of depths. A systematic study of these fossils and its occurrence revealed that the deepest layers were found to have fossils of invertebrates. In layers above them were found prehistoric fish-like animals, amphibians, reptiles, birds and mammals respectively in that order. This suggests that perhaps this was the order in which the animals appeared on the earth. It is interesting to know about connecting links. Organisms referred to as connecting links are those which are structurally intermediate between two different groups. Connecting links can be found among organisms that are still living. Example, Peripatus has segmental nephridia, thin cuticle and parapodia-like appendages as in Annelida. At the same time, it has trachea and open circulation as in Arthropoda. The duck-billed platypus lays eggs like reptile and has hair and mammary glands like mammals. Lungfish, though a fish, breathes air through its lungs. These organisms point strongly to the fact that mammals have evolved from reptiles and amphibia from fishes. Embryological evidence. Embryology deals with the study of development of an organism from an embryo. The comparative study of embryos of different generation of vertebrates exhibits certain interesting facts. The embryos in their early stages of development show a great deal of similarities, but differ in later stages of development. This suggests common ancestry for these animals. Let us know more about Charles Robert Darwin, FRS, 12th February 1809 to 19th April 1882. He was an English naturalist who established that all species of life have descended over time from common ancestors and proposed the scientific theory that this branching pattern of evolution resulted from a process that he called natural selection. He published his theory with compelling evidence for evolution in his 1859 book on the origin of species. The scientific community and much of the general public came to accept evolution as a fact in his lifetime, but it was not until the emergence of the modern evolutionary synthesis from the 1930s to the 1950s that a broad consensus developed that natural selection was the basic mechanism of evolution. In modified form, Darwin's scientific discovery is the unifying theory of the life sciences, explaining the diversity of life. Let us study Darwin's theory of natural selection. On the basis of the observations, Darwin suggested that only the fittest survive. All those plants and animals which are not fit die. These fit species reproduce and pass on the relevant characteristics to the following generation, which in turn would make them fit for survival. The process of selection of characteristics that contribute to the fitness for survival was called natural selection by Darwin. Why is there a competition between individuals of the same type? There are many differences found even in the individuals of the same species. Some individuals have certain factors to a greater extent than others. If these factors are necessary for survival, then these individuals can survive the struggle for existence. Other individuals who do not possess these factors die. These useful factors appear only in some individuals in the next generation of the species. Here also, in the struggle among the individuals for survival, only those possessing the factors at greater levels survive. This process continues from generation to generation. The total effect is that after several generations, the number of individuals having relevant factors that are better adapted to their surroundings is much more than in previous generations. These adapted individuals may also be very different from the original species. This process is described as natural selection of these individuals which have characteristics best adapted for survival. Selection by nature is not deliberate but is natural. The criterion for the natural selection is only one, that is, successful adaptation for growth and reproduction in the given environment. The natural selection of the individuals best adapted to their environment will be made clear from the example. A certain variety of moths which often fall prey to birds is found in the surroundings of Birmingham in England. These moths are found in two colors, black melanized and grey. Both these varieties live on the tree trunks. Years ago, trees in this particular area were hosts to the growth of algae and fungi and thus were grey in color. Hence, the grey moths were easily camouflaged on the tree trunks and were protected from their predators. The black moths were easily noticed by the predatory birds and were consequently eaten by them in greater numbers. So the population of grey moths was much higher than the black ones. Later, as a result of industrial revolution, there was an increase in the level of pollution. Owing to this, there was hardly any growth of algae or fungi on the tree trunks. Instead, the tree trunks got covered by soot, which gave them a black color. Now, it was the black moths that began to get protection, and the grey ones were easily detected by their predators and consumed by them. Nowadays, in the surroundings of Birmingham, black moths are found in large numbers. The theory of natural selection, which was proposed by Charles Darwin, helped to explain the process of the development of living things. However, the theory did not explain how an individual plant or animal acquired factors that made it better adapted to its surroundings. In the course of time, these questions were answered by the discovery of the laws governing heredity and mutation, and thus Darwin's theory came to be universally accepted. Let us understand the term Lamarckism or Lamarckian inheritance. This is the idea that an organism can pass on characteristics that it acquired during its lifetime to its offspring, also known as heritability of acquired characteristics or soft inheritance. It is named after the French biologist Baptiste Lamarck, 1744 to 1829, who incorporated the action of soft inheritance into his evolutionary theories. He is often incorrectly cited as the founder of soft inheritance, which proposes that individual efforts during the lifetime of the organisms were the main mechanism driving species to adaptation, as they supposedly would acquire adaptive changes and pass them on to offspring.